Okay, are there any questions? Let me show you where we're starting. So today we're going to be talking about Lab 5, Streaking for Isolation and Culture Media. Sometime this week, probably not until now, not until Thursday, but you do need to read the unknown projects instructions on Canvas for next week. I'm going to be starting the unknown project, and I will discuss it, but I want you to read the unknown project instructions before I begin discussing the project. I think I already talked about the quiz. I forgot to mention, let's go briefly to Canvas, that I graded, I think 3 or 4 assignments in Canvas. One of them was the discussion in the lecture, and there were at least 2 labs that I graded. I might have graded even 3. Over the weekend till, I don't remember, maybe last night. And you have until Thursday to update your answers. After Thursday, you will not be able to update your answers. So you have until 11:59 p.m. on Thursday to update your answers. And like I said, I started that. Actually, I think I started it on Saturday. It was one of the labs I hadn't gotten graded, and it wasn't due this Saturday, it was due last Saturday. All right, let's go through and just make sure I'm not missing anything. Don't forget that if you want an extra credit study, an extra credit point, get together in a study group, click on the link because it'll tell you what you need to do to get the extra credit point. And then one person in the group needs to submit it. There is a quiz tomorrow, both Quiz 2A and Quiz 2B tomorrow is due. All right, so we'll talk about the lab module 5, and remember to submit your answers on the worksheet, and then only upload the worksheet. The worksheet is much smaller in size than the lab module. Generally speaking, it'll have the same questions, but the questions could be different, meaning that if I decide the question wasn't quite right, I would make the changes in the worksheet first, and then upload that. Okay, any questions? We are recording. All right, so let's move on. I think I don't need that anymore. So let's talk about Lab Module 5, Cultivation of Bacteria, Culture Media, Inoculation, and Isolation. Of course, pre-read the textbook. The reading isn't long this chapter. There will be video clips, and I will look and see if there's a virtual lab I'm sorry, I should have looked that up. I have a feeling that the virtual lab for this one uses Flash, and if it uses Flash, you won't be able to do it. Primary learning objectives. Upon successful completion of this laboratory, students should be able to understand the importance of proper specimen collection and transport to the laboratory. Explain the 5 principal steps used by microbiologists to process specimens. Differentiate between the various types of culture media. Explain the purpose of streak plate culture technique, meaning streaking for colony isolation. Define the following terms: selective media, differential media, and reducing media. The ability to isolate pathogens from a clinical specimen submitted for culture is only as good as the collection technique and the timely transport of the specimen to the lab. Some microbes are sensitive to desiccation, drying, and exposure to oxygen, meaning anaerobes in particular, and require a special transport medium. Once the specimen is received in the lab for microbiology bioculture, microbiologists employ the 5 principal I steps to process it: inoculation, isolation, incubation, inspection, and identification. And so 1, inoculation; 2, isolation; 3, incubation; 4, inspection; 5, identification. Inoculation is placing the specimen on an artificial culture media to initiate the growth of the desired organism. Isolation will be spatially separating microbial growth into distinct colonies, and then isolating the colony of interest. Incubation, placing culture medium in an environment where conditions facilitate the growth of the bacteria. Inspection, examine the culture for the presence of microbial growth. And then identification, perform specific tests to ID any specific pathogen that is isolated. The 5 steps we'll discuss in a little more detail, and they use different types of culture media. You must know the different types of culture media. We've talked only about one type of media, and that is a general purpose media. You're familiar with those. Nay, nutrient agar, nutrient agar with yeast extract. I don't know how you pronounce this. Nay, and this is sort of Na for nutrient agar. And then TSA, tryptic soy agar. These agars allow you to grow many bacteria, but not all bacteria. For example, fastidious organisms will not grow on a general purpose media. To promote the growth of fastidious microbes, those that have very specific nutritional requirements, you have to use enriched general purpose media. For example, 5% sheep's blood added to the media can help an organism grow. I'm trying to remember, Streptococcus pyogenes has to grow on blood agar or some other enriched general purpose media. It cannot grow on a general purpose media. E. coli can grow on a general purpose media. All right, so those are 2 media. There's also a selective media. A selective media contains chemicals that inhibit or halt the growth of some organisms. Keep in mind 2 things when using selective media. First, a selective media isn't necessarily perfectly selective. meaning the organism may grow, just not as quickly as it would on a general purpose media. And second, even those organisms which are supposed to grow may grow smaller colonies or grow more slowly on the selective media than they would on a general purpose media. There are many kinds of selective media. An example of a selective media is a 7% sodium chloride. This selects for organisms which can grow in 7% sodium chloride. Most organisms cannot grow in 7% sodium chloride. All right, any question about that? All right, differential media is a media that contains chemicals that allows you to see visible differences among groups of different organisms when they're grown on that media. These differences are unrelated to how well the organism grows on the media. Different culture media allow dissimilar bacteria to be visually distinguished from each other, so they will look different on a differential media. What is a differential media? It is only differential. I can't think of one that is only differential and nothing else. So I'll just say McConkey's is a differential media, and it allows you to distinguish between cells' differences to metabolize the sugar lactose. If they ferment lactose, they will be pink on McConkey's media. If they don't ferment the sugar lactose, then they will not be pink. They will be off-white or some other natural color that the bacteria has. There's one more type of media you need to know. It's a reducing media. A reducing media is one that promotes the growth of anaerobic bacteria. What a reducing media has are chemicals that remove oxygen from the media. So there will be either low to no oxygen in the reducing media. And anaerobes will need to grow in a reducing media. Thioglycolate is a reducing media, and we'll talk more about thioglycolate later. All right, it's important to note the culture medium can be classified into more than one category. For example, McConkey's is both selective and differential. And blood agar is both an enriched medium and a differential medium. Here we're looking at growth on a McConkey's plate. It's selective in that only gram-negatives can grow on the McConkey's plate. And both of these are gram-negatives. It's differential in that if the bacteria can ferment the sugar lactose, the bacteria will appear to be pink colonies. If the bacteria does not ferment the sugar lactose, the bacteria will not be pink. McConkey's, the reason why it's selective is that it has a pH indicator which turns pink when it is acidic, and it becomes acidic if it ferments the sugar lactose, and it's not pink because it's not acidic when it does not ferment the sugar lactose. It's selective in that it has chemicals that do not allow gram-positives to grow. McConkey's has dyes in it. Let me see if I can remember the dye. Sorry, I'm drawing a blank there. It's something very common, like bromopheno blue. Maybe that is it. In it, as well as bile salts. And gram-positives don't like growing in bile salts. Gram-negatives are fine growing in bile salts. And is it crystal violet? I can't remember. I think it is crystal violets in McConkey's. That's why it's purple. When you look at the plate before growth, it's hard to look at that now because we have growth on it. But the dye gets in the cell wall of the gram-positives, and the gram-positives don't like that dye in the cell wall, and it prevents the cells from growing. So it's selective on gram-positives, but gram-negatives are allowed to grow on it. Here we're looking at blood agar, and it is a enriched media. The blood provides the nutrients for Streptococcus species to grow, like Streptococcus pyogenes will not grow on a general purpose media. It's also differential in that we can see differences in the hemolytic patterns of the bacteria on a blood agar plate. You can have no hemolysis, also called gamma hemolysis, and that's right here, where there's blood up until the bacteria cells. In fact, there's probably blood underneath the bacteria, but we can't see it. And then there's alpha hemolysis, where there's blood, but you see there's a dark ring around the bacteria, and then there's a dark ring, actually it's completely dark inside the bacteria. That's dark because of partial hemolysis of the blood. The blood has been partially hemolyzed, lysed, and so it appears dark. And then there's beta hemolysis, or complete hemolysis. And here we have the blood, then we see a dark ring. That darkness is actually alpha hemolysis. And then surrounding that we have a clearness around the bacteria, which is in the beta. And that clearness is because the bacteria have been completely hemolyzed. And so you can look right through the plate. Okay? Usually when you have beta hemolysis, you'll have a ring of alpha hemolysis around it. All right, any questions about blood agar plate? So you need to know this table, looking at the different types of media, the general purpose media, the names of them, the differential and enriched media, which really is on the blood agar plate. This is an enriched general purpose media. It's chocolate agar. Chocolate agar is the same as blood agar, except you add the blood while the agar is still very hot. Meaning you took the agar into the autoclave. You took the media into the autoclave and you add the blood at that point, and the blood lyses, and it makes the agar turn brown, which is why it's called chocolate agar. With blood agar, they allow the agar to cool, and then they add the blood while the agar is cooled, but before the agar solidifies. Okay? Any question about that? The important part is those are two enriched general purpose media. And blood agar is also a differential media. Let's move on. There is only one selective media that we're going to talk about. That is phenyl ethyl alcohol. It's PEA. It's used to isolate gram-positive bacteria. It has phenyl ethyl alcohol in it, and it selects against gram-negatives. The phenyl ethyl alcohol gets in the outer membrane of the gram-negative cell and prevents them from growing. So PEA is a selective media. We then have a number of selective and differential media. MacConkey's, MAC, eosine methylene blue, EMB, and hectone agar, HE, are all selective and differential media. They're selective in that we use them to grow gram-negative bacilli. They select against gram-positives. They're differential on their basis to ferment the sugar lactose, and they'll be a different color depending on whether they ferment lactose or not. The difference is that each one will be a different color. MacConkey's, I remember, because I've done it so many times, it's pink. I think on hectone agar, if it ferments the sugar lactose, I think it's orange or yellowish. And then with EMB, it will be greenish or metallic, like metallic black color on EMB, if it ferments the sugar lactose. Mannitol salt agar is also a selective and differential media. It is selective in that it has a high salt concentration, so only organisms that can tolerate the salt can grow. It's differential in that it allows you to differentiate on how the species can ferment the sugar mannitol or not. If it can ferment the sugar mannitol, it'll be yellow in color. If it does not ferment the sugar mannitol, it will be non-yellow. Usually it will be off-white in color, but it depends on the species of bacteria. The important thing is not yellow. Mannitol salt agar is frequently used to distinguish Staphylococcus aureus from other staphylococcal species, because Staphylococcus aureus can ferment the sugar mannitol, and Staphylococcus epidermidis cannot. Most staphylococcal species cannot, but there is one staphylococcal species, and I'm forgetting the name of it now, can ferment the sugar mannitol, as well as Staphylococcus aureus. All right, isolation of bacteria was first started by Louis Pasteur. You don't need to know the year he did it in, but he came up with the concept of a pure culture. His way of isolating a pure culture was actually very difficult. He had to dilute the bacteria, and you had to dilute it a lot. And what he did was he diluted it to a solution that if only 1 in 10 tubes had bacteria growing in it, he said, well, that's probably a pure culture. Okay? Because 9 out of 10 times I didn't get any bacteria growing, it was so dilute. Anyways, he developed the concept of working with the peer culture, and his mechanism for getting it was not very easy. Robert Koch, you don't need to know the year, but in 1880, developed a much easier way of getting a peer culture, and that is you streak it out on an agar plate for colony isolation, meaning you're streaking for isolated colonies. And if you do that 2 times, meaning you then go and once you have an isolated colony, you then pick up bacteria from one colony and streak it out for colony isolation on a second agar plate. And if you've done it 2 times, you have a really, really high chance. It's greater than 95% chance that you have a pure culture. Anyways, Robert Koch was the first to develop growth on a solid medium and streaking for colony isolation. And that was much easier to get pure cultures. So here we're streaking for colony isolation. There's quadrant 1, quadrant 2, quadrant 3, and then quadrant 4. You can notice that there's a high concentration of bacteria in quadrant 1, and then it's a reduced number in quadrant 2, reduced in quadrant 3, and then in quadrant 4 it was reduced to numbers low enough to get isolated colonies. It doesn't matter which quadrant we get isolated colonies in. The point is you just want the bacteria numbers to be reduced to get isolated colonies. To get a pure culture, like I said, you would then take from this colony and streak it out again on an agar plate. And then get isolated colonies, and then to say it's a pure culture, grab from one isolated colony. So you need to go through 2 isolated colonies before you can say it's a pure culture. Incubation is just to grow the culture in an environment that's necessary for it to grow. Most of the nutrients will be supplied by the growth medium. You just have to choose the correct medium. The environment is what we incubated in, usually called an incubator. It could include things like changing the oxygen and the CO2 availability, the temperature, the humidity, and of course the time. Our incubators are very simple, and all they do is we use the time, the humidity, and the temperature. The oxygen and the CO2 simply come from the air, so we don't change it. Then you inspect the culture to make sure you have visible colony, visible isolated colonies, and you see morphological variations between different species of bacteria. Microbiologists can look at these differences and make some characteristics about the species from the colony characteristics, like the size, the shape, the margin, the elevation. For example, if you had a beta hemolytic bacteria on a blood agar plate, and it was a very small pinprick of a white colony, small pinprick in size, the margin would be entire. You're likely to conclude that that's Streptococcus pyogenes if you did a strep throat what do you call that, a strep throat test. Most of the time, though, you'll need to do identification, and you'll take that species, you'll need to make it a pure culture, and then run biochemical testing on. And we'll talk more about that in the later labs. So know the terms of this lab. And we have some videos for you. You only need to watch the video to 4 minutes and 15 seconds. We're looking for streaking for colony isolation. And then look at the videos on beta hemolysis, alpha hemolysis, and gamma hemolysis. Another video on McConkey's agar. You only need to watch until 350. And then a video on mannitol salt agar. And this virtual lab you don't need to run. You won't be able to get it to run because it requires Adobe Flash. And my apologies, I'm working on removing these from the lab, and I just haven't gotten to lab 5 yet. I think I'm working on lab 4 now, and I stopped to get the homework done, and I need to get back to this project. So you won't be able to do the virtual lab, so don't try. And then answer the questions in the worksheet, not on the lab manual. You need to answer this question, fill in the table with X's, and then answer question 9 as well. And then submit your answer to the folder on Canvas for Lab 5. All right, any questions?
Does that mean that we'll find the answer to those questions in the videos that you're saying to watch?
Some of the answers will be in the videos. Most of the answers will be in this lab manual.
Okay.
Okay? All right, I'll be on until 7:30 to answer any questions.